Great pleasure to, okay, another pop-up. All right. To speak at this opportunity today. So we're going to continue the conversation that Thomas has started, how we explore the ever-growing large chemical spaces for novel heat finding. There are many heat finding technologies in the modern drug discovery space. Here we list a few provisions that WuXi AppTec has been using to service our customers globally. This include experimental screening technologies that are high throughput screening assay enabled, as well as small molecule or fragment-based screening based on biophysical methodologies. On the larger scale of the experimental side, we have DEL screens that allows us to prosecute billions of compounds. In complementary to all these experimental technologies, we're going to focus on today on the virtual screening or computational approaches for heat finding, which we will learn that they, unlike the experimental methods, which are optimized toward a specific target class or could be amenable for certain types of chemical matters, the computational approaches are more general and more agnostic to the specific target class and the modalities that are of interest. So could be of more utilities as we grow in the power and the algorithms in enhancing those computational methods. The computational approach or artificial intelligence approaches has a deep roots in drug discovery process. In the different stages, we see different technologies being employed from early time target identification and validation through compound screening all the way to preclinical or clinical developments. At different phases, different technologies are employed, different problems are asked and being solved. This include, early target ID space. We utilize gene disease associations and splice variants information to prioritize the targets and maybe the druggability predictions. During the heat finding stage, we employ the synthesis, planning, and reaction predictions, also in combination with a lot of property predictions to prosecute and accelerate the heat finding and advancement to lead optimization stage. At different phases, different data were required to support the different methodologies to problem solving. And there are apparently a lot of gaps in those spaces as well in terms of data homogeneity, the curation quality of the data, and also the access of the sharing of data for different applications. Nonetheless, given the vast growth and development in the software and hardware space, the artificial intelligence or computational intelligence has been a indispensable component in our discovery endeavors. Just to further support that notion, here's a graph of all the activities of AI in drug discovery publications. graph on the left, and also the venture capitalist activities on an annual basis in the recent past. As we see, the growth had seen the exponential increase, and this tracking activity only stopped at early 2020, as the author Simon Smith put it. um, I have to stop because I can no longer keep up with the developments in the use of AI in drug discovery. So we are really seeing a huge surge in the technology uh, utilization these days. Virtual screening is a computational technique to search in real or virtual libraries of small molecules in order to identify potential hit candidates. Here we use a small molecule as a example, but the technology can be easily expanded to include other therapeutic modalities, such as peptides or even oligonucleotides. So the libraries of input can be either real or virtual, which ranges from hundreds of thousands to millions of compounds, or the virtual library. In the virtual library case, it's ever growing. The WuXi Galaxy currently provides about 3.5 billion compounds to prosecute. We then utilize different technologies to evaluate the likelihood of those compounds um, in terms of their match to our specific therapeutic target. This could be structure-based methods or could be pharmacophore or ligand-based approaches. When the protein structure information is available, it's the most advantageous to employ the docking algorithms to accurately assess the binding complementarity of the compounds with the protein. And different screening or secondary filtering strategies can be employed as well in this large funnel. This could include property filters, including ADME properties, CNS penetration, or even some tox considerations, and could be some specific chemotype that we would like to focus on. So in the end, we get a list of hit candidates that we can submit to biological assay to confirm their activities. In this paradigm, different methodologies has different accuracies, as Thomas also illustrated in his talk, and the different time requirements. Docking-based methods are on the more expensive side, followed by pharmacophore models and shape screening. Fingerprint methods are much faster, and especially the more modern machine learning-based screening technologies, which really will allow us screening huge number of compounds in a very efficient manner. So we'll talk more about that as we move on. There are a number of commercial catalogs that can be utilized for the screening campaign. They provide different categories of drug likeness, different properties of the compounds, and they serve as a good source of quick access to some initial chemical matters for testing. Um, so it is a attractive place to go look for the commercially available information when we start with a particular target screening. And in a hypothetical sense, this is the a possible outcome from a virtual screening workflow. So we started with 2 million compounds at a very high throughput screening mode and funnel that down to the higher precision docking methods at about 10% retaining rate. In the end, we have a few hundred to thousand compounds that we can submit to our biological assay, depending on the throughput of our testing scheme. The distribution histogram on the right shows us the propensity of compounds at the different scoring windows. Toward the left-hand side are the compounds that are predicted to be higher binding toward the target and are of more interest to us. So we may want to select, 2 standard deviations away from the mean to for the curation for the biological assays. On top of, or in addition to the real chemical matters or commercial compound collections, we also have access to the make-on-demand libraries. This size is ever growing at a very fast rate. It is predicted that we are operating in the billions range, and we are growing in that space at an exponential rate. In addition to the size or the number of the compounds, we are also growing in the diversity or chemical representation of different matters based on the number of novel scaffolds that we have employed in building those make-on-demand libraries. So along with the increase in the scale, we are also enhancing our coverage of the chemical space by introducing novel chemical scaffolds. Using the docking-based methodologies, the scientists at UCSF, in collaboration with other research labs, has demonstrated great success to identify heat and very potent lead matters in large compound collections. This is a reference that Thomas has touched on as well. On 2 separate targets, MPC beta lactamase, as well as the D4 receptor, the totally different chemical classes, or I'm sorry, biological classes. The researchers has demonstrated very impressive successes in identifying lead compounds. In the beta lactamase case, only 44 compounds were synthesized, and a novel phenolate chemotype were discovered and soon was quickly optimized to a 77 nanomolar inhibitor via very few rounds optimization. In the D4 receptor example, they tested a little over 500 compounds and found 30 new chemotypes, all of submicromolar activity. And the most compound showed picomolar D4 agonist activity that is subtype selective. In the following data analysis, they also demonstrate that the hit rate is highly correlated with the docking score. So in this graph here, toward the left-hand side, the lower docking scores, the hit rate is at about 26%. As we move toward the x-axis in the direction of worse docking scores, the hit rate apparently monotonically decreases all the way to 0. This is a very strong evidence that we can have predictive power in the large, diverse compound collections using computational methodologies. Also, they did an interesting comparison between machine selection and human selection in the final hit candidate. So in one case, the medicinal chemists were provided with the docking results. So they were given the opportunity to provide expert opinion to make the final selections. And the comparison produced results that the two selection mechanisms yielded very similar hit rates. Not only we can predict the binding affinity or identify binders from large decoy compound collections, the binding poses were also shown very high fidelity to the experimental structures. So out of the hits identified from this virtual screening campaign, a few compounds were later resolved by X-ray crystallography, and the experimental poses showed remarkable consistency with the docking predicted poses, as evidenced by both the key interaction elements, the orientation in the binding pocket, and most importantly, the root mean squared deviation, um, which are all very low in these instances. So not only we can predict the affinity, but also the actual correct poses of the ligand protein interaction. In addition to docking-based virtual screening methodologies, we are also seeing a lot of applications of machine learning endeavor in further expanding that chemical space and the speed of prosecuting large compound collections. So in this machine learning empowered virtual screening place, we can provide rational starting points, also evaluate target binding with pretty high success rate of activity, and even apply the generative approaches of the machine learning algorithms to further explore or expand the chemical space. In this seminal paper, the group of scientists at MIT took on the endeavor of identifying novel antibiotic agents using the existing data that were designed for the screening campaign, as well as other sources. They developed this sophisticated deep learning based neural network models and apply those models to predict the of billions of compounds, hundreds of millions or close to billion compounds to look for the novel chemotypes. And interestingly, they did find something that is new or unknown to the antibiotic space. Not only the model were able to predict the chemically related compounds to demonstrate the antibiotic or growth inhibition in their assays, but also the unknown chemotypes that were newly discovered. And this new chemotype was able to demonstrate efficacy in the in vivo infectious model later on. Another example utilizes the combination of machine learning and docking to lead finding. This is similar to what Thomas has talked about, sort of the bootstrapping way to generate the data that's needed for machine training, as well as the model refinement and prediction using computational approaches. So for 12 different targets, the deep learning platform was used to screen over a billion compounds, and a subset of the initial compound collection were docked to generate the scores um, for the subsequent training of the machine learning models. And this resulted in 100-folds of data reduction and another few thousand folds of enrichment of high scoring molecules. So when the experimental data are not readily available, um, the machine learning model can be um, applied together with the docking algorithms or traditional computational or CAD approaches to generate and utilize the information to enable the faster processing of large amounts of data. The structure-based virtual screenings relies on the accuracy and representation of the protein models to enable the exploration of different conformational states. MD simulations are sometimes necessary to explore the conformational flexibility. In this membrane protein system, we embed the protein into the lipid bilayer, saturated system on both faces with water, and add the ions and at physiological conditions to simulate the biological environment. And after the sufficient long time sampling, we can analyze and cluster the different conformational states and select the most representative conformations for our docking exercise. So this allows us to capture the more diverse and representative and accurate protein conformations in our virtual screening campaign. At the time when we are trying to apply different methodologies and improve on our algorithms and resource capabilities to increase the execution of chemical spaces, we're still looking at a very local or limited area, as the estimated chemical space is the size of the universe. It's not something we will ever come close to fully explore. So one area of interest is to explore in a local or specific chemical space that are defined by either the protein target or the chemical series of interest, and use the generative machine learning models to generate analogs around that chemical space and further explore that fine grid of chemical representations. So in this instances, we are really trying to cover or design around a very well-known defined chemical space, and that allows us to apply some of the more exploratory and accurate methods to more accurately understand that space and be focused on the areas of interest. So this is an iterative process. Once we have the target chemotype, we can generate analogs using deep learning methods using that initial lead and submit the outcomes from those generative models to downstream evaluations, which could include structure-based screening and other ligand-based approaches. And this process can iterate for several rounds to continue to train the model and zoom in the most productive areas of analoging. A step further from generating our virtual screening hits is converting those hits to lead and optimizing their properties for candidate selection. So in that space, we also have a number of machine learning based algorithms to help us accomplish those tasks in the efficient and effective manners. The deep learning models can be built on molecular fingerprints or feature trees or graph representations, even on the smile strings. Uh, the ultimate embedding process of that different representations gives us a deep, complex neural network architecture that allows us to build connections between different layers that can identify the hidden connections or correlations between the input and output. We can accurately develop models to predict properties of importance, such as binding affinity, selectivity, or other ADME properties. Or we can use these models to provide analoging ideas. So all of these approaches will take us one step further from the initial hit finding stage to the lead optimization. Checking on time. And at WuXi, we have a technology platform that supports the activities around the design, synthesis, test cycle for the lead generation. And at different stages, we generate the necessary data from the experimental technologies and combine those data with the powerful analytics tools to distill information and inform knowledge to our next rounds of design and experiments. This permeates through the early phase hit discovery, synthesis planning, as well as the pharmacology evaluation in our technology platforms. So in summary, I have given you examples of different computational technologies, how they can be employed in the hit finding stage, which is a very critical phase of drug discovery research for chemistry initiation. And every technology has its role in this streamlined approach. We hope to employ, all the tools in our toolbox and develop the new approaches to enable the efficient, accelerated execution of our drug discovery research. With that, thank you for your attention and happy to join the discussion.